It is a topic on unique and repetitive DNA sequences, mini-satellites and microsatellites. If genome is annotated, we may get different groups under it. Basically, a genome can be divided into two groups, that is coding and non-coding sequences. Under coding sequences, there are genes which synthesize RNAs and thereafter proteins. Non-coding sequences can be further divided into two groups as repetitive DNA and introns, regulators, etc. Repetitive DNA can further be divided into two groups as tandem and interspersed. Tandem sequences are satellite DNA sequences which may be of mini-satellite and microsatellite. Interspersed repetitive sequences may be of different types like SINE, LINE, LTR, transposons. Unique sequence of DNA is a kind which represents the genes. These are the sequences of nucleotides which are generally present as a single copy or only a few copies within a genome. It represents almost 55% to 60% of the entire genome. Repeated sequences are those sequences which occur in multiple copies within a genome. It makes a significant fraction of the genome DNA, which is almost 2/3 part of the human genome. Repeat sequences may be categorized into different classes based on their structure and mode of multiplication. The very two major classes under these are tandemly repeated sequences and the repeats which are dispersed throughout the genome. Tandemly repeated sequences are also known as satellite DNA. It is being called as satellite DNA because while extracting the entire genome from the cells of an individual, after centrifugation of the extracted DNA in a gradient medium, two or more than two bands appear, one of which is a very thick band representing the most part of the genome, while the another band represents this satellite. This satellite appear as the representation of repetitive DNA, tandemly repetitive DNA. In this picture, you can see how the satellite bands and the major band or the main band are appearing along the length of the gradient gel. This picture shows the graphical representation of the main band and the satellite band. So, types of repeats are two: Basically, these are tandem repeats and interspersed repeats. Under tandem repeats, the satellite DNA may be mini satellite or microsatellite. Mini satellites are the repeat units which vary from 10 to 60 base pairs in length and found in many places in the genome, including the centromeres. Whereas microsatellites repeat units are comprised of less than 10 base pairs. These include the telomeres. Interspersed repeats are distributed in a scattered manner throughout the genome. This kind of repeats may be of transposable elements, which may be transposons or retroposons, LTR retroposons, non-LTR retrotransposons, then short interspersed nuclear elements, long interspersed nuclear elements, all these things. In primates, the majority of the lines, that means long interspersed nuclear elements, are line 1, and majority of signs are ALU. In prokaryotes, CRISPRs are arrays of alternating repeats and spacers. Here are some other types of repeats classified under two major groups: direct repeats and inverted repeats, as well as mirror and everted repeats. You can see here the examples of direct repeats: simple repeats, inverted repeats, palindromes, mirror and everted repeats. Here again you may get a further illustration of single copy DNA or the unique sequence of DNA. Under the kind of repetitive DNA sequences, some repeats may be of moderate nature by their presence, which vary from 10 to 10 to the power 5 copies per genome. They are found throughout the euchromatin. Average size of each of them are generally around 300 base pair. They may be further classified as microsatellite or mini satellite, example of which may be like variable number tandem repeats, that is the VNTR, and the second group may be dispersed repetitive DNA, mainly transposable elements like lines, signs, etc., and also includes redundant genes for histones and ribosomal RNA and proteins also. Some of these repeats are also involved in the regulation of gene expression. This notion is supported by the evidence of their presence, the presence of these repeats interspersed with, that means mixed with, single copy sequences, as well as their location very much adjacent to the structural genes. Highly repetitive DNA sequences, which represent 10% of the total, occur more than 10 to the power 6 copies per genome. Their individual length are rather short, which varies from 5 base pair to 100 base pair. Most of these repeats are present at the heterochromatic region of the genome, like around centromere or telomere. Their possible function includes structural or organizational roles, and they are also functioning in the chromosome pairing. They are involved in the crossing over or recombination, or may be present just as the junk. You can see here a further illustration of microsatellites, mini satellites, and highly repetitive DNA. Here you can see a comparative account of unique or single copy DNA sequence and repetitive sequence. Transposons. Moderately repeated DNA are mostly transposons. Two major families under it are long interspersed elements, that is lines, and short interspersed elements, that is signs. L1 is an example in human of the first kind, that means under long interspersed elements, and ALU element is an example of signs. Both of them are retroposons. These elements play a significant role in genomic function and evolution. However, majority of such elements are not in their full length. Some of them, the majority of them are truncated, that means cut into short part. They are not present in the full length. L1 and ALU elements are apprehended to be developed by retrotransposition. L1 in human beings, an example of lines. It repeats almost around 50,000 times in human genome, comprising almost 0.5% of the total. However, out of them, merely 3,000 copies are of full length, and rest are truncated, that means short, not having the full length. And this truncation is taking place mostly at the 5' end of these elements. Full length of such element is of 6 kilobase pair length with two open reading frames. Two open reading frames means they are having two sequences which can be read for different meanings, for different functions, one of which encodes reverse transcriptase. Near the 3' end of this element, the sequence is very much AT rich. This element is flanked by two short direct repeats. ALU family is named after a target sequence which is present within this sequence for the restriction enzyme ALU1. This repeat occurs as 50 to the power 5 to 50 to the power 6 copies in haploid genome. Almost as a repeat in every 4 kilobase pair, and so varying from 1 to 10% of the total. They generally occur within introns non-translated regions of the mRNA and not encoding regions. It has 209 base pair consensus sequence comprised of 2 tandem repeats of 130 base pair, one of which has again 32 base pair deletion. These elements are flanked by direct repeats. I mean, on both sides of them there are direct repeats. Each unit of this repeat is with an AT-rich region which suggests a poly A tail. 5' end is very much similar to polymerase 3' promoter region. Both of lines and signs have poly A tail regions. These regions are supposed to act as template during reverse transcription from nicks, which nicks are made at the position of the DNA sequence where these lines and signs are to be inserted in the host DNA. This insertion is being facilitated by line encoded endonuclease. But one difference is in case of ALU sequence, which is transcribed but not translated, because the insertion of this ALU, I mean the transposition of this ALU RNA, might be done with the endonuclease or reverse transcriptase encoded by L1. Transposons are also involved in diseases. Retrotransposal integration of ALU and L1 sequences within important genes play significant roles in some human diseases. However, a very small number of transposons, which is even less than 0.01%, can transpose and may cause mutations. However, mutagenesis by signs and lines by getting inserted in mammals is becoming more abundant. L1 in particular has been found to be involved in different maladies like hemophilia, DMD, breast and colon cancer. They are involved. Though the exact mechanism is still unclear about their role. Evidence shows ALU sequences are being integrated in the very control regions of genes, that means the promoter enhancer regions of genes, and being positioned in those regions, these sequences, the ALU sequences, they can modulate the functioning of the genes, the transcription of the genes, by binding some regulatory proteins there. ALU sequences show variations in their sequences and thus making a family. It occurs due to unequal crossing over between repetitive elements causing gene duplication.